Understanding climate change in East Africa and Southern South America through marine fauna fossil records. Hello, my name is Omar Sarate, and I will be presenting how marine fauna has helped scientists and climatologists reconstruct ancient ecosystems and climate change in two specific regions. To explain how marine fauna paleoproxy records are used to record changes in climate, I will be referencing the research that is covered in bibliographies A and B. Marine fauna paleoproxy records are freshwater organisms that have fossilized and preserved in sediment layers at the bottom of lakes and rivers. These include macrofossils like fish fossils and microfossils like foraminifera and mollusks. These organisms act as environmental archives and record water temperature, salinity, or sea level changes, for example. Figure 2 provides us an overview of the study strata in the Tugan Hills of East Africa that describe the layer type compositions, layer depths, and depth of found fish fossils. We will first look at the sedimentary records from East Africa during the middle to late Miocene, about 15 to 10 million years ago. In the Tugan Hills of the central Kenya Rift, freshwater lagerstätte from the Ngoro Formation suggests the previously humid and forested paleo environment. The research of sediment cores, taxonomic identification of fossilized fauna, and paleontological analysis provide evidence to past lake conditions. Radiometric dating further provided paleoecological data that indicated an increase in the decline of marine biodiversity in Poland. This regional drying is interpreted to result from a combination of tectonic uplift and global cooling events, which disrupted precipitation patterns and altered the hydrological balance. Figure 3 shows us the articulated fish fossils discovered in the Angora Formation. We will now look at records from the Pleistocene about 125,000 years ago in Navarino Island of southern South America. Microfossils like foraminifera and ostracods found in uplifted marine terraces provide evidence for elevated sea surface temperatures and marine transgressions. Researchers relied on sedimentological sampling and stereoscopic and microscopic analysis of marine fauna fossils to pull data regarding region temperature and water chemistry. Stratigraphic data, along with oxygen and carbon proxies pulled from calcium carbonate shells, example shown in Figure 4, provided the conditions of the paleo environment reconstruction of the Navarino Island about 125,000 years ago. Consistent with global interglacial trends, the findings in this region revealed a marine environment that adapted to a warmer climate. The fossil assemblages discovered serve as reliable indicators of sea level fluctuations and regional climate regimes. The current climate in both regions can be described in terms of the global wind circulation patterns. The central Kenya drift is located within the Hadley cell. This region is tropical and semi-arid climate. Seasonal shifts in intertropical convergence zone can influence monsoon behavior and increase drought risk. On the other hand, Navarino Island is located near the subpolar Ferrel cell. The climate consists of cool temperatures influenced by the westerly winds of the southern hemisphere. Variability in the wind patterns can influence precipitation in the region and alter sea temperatures. In conclusion, marine fauna fossil records offer a dynamic understanding of long-term climate change. With techniques like isotope chemistry and marine fossil analysis, we observed in the central Kenya drift the shift from a humid region to arid conditions over a period of millions of years. In southern South America, marine fauna records captured the thermal and coastal characteristics of the last interglacial period. Understanding the climate's past change is essential to our planning for the future. Marine fossils tell stories of changing climates, and understanding those stories is key to predicting and preparing for the future. Thank you.